Everybody. Thanks, Hilal, for accepting the invitation. It's a great pleasure to have you here. I think that in the field of aggregation, Hilal Lashuel doesn't need much of an introduction. And as you know, the introductions here in these webinars are always short. He has contributed so much in our present understanding of protein aggregation in vitro and in vivo, and characterization and the comparison between what happens in vivo and in vitro and the relevance for each other, that I don't want to spend more time to describe this, because also from what he told us, this is going to be what he's going to tell us about challenging also our view of what we do in vitro, its relevance in vitro, in vivo, and so on. So with this, I keep it short. Hilal, the virtual floor is yours.
Thank you very much, Paolo. Thanks for the opportunity to give a talk and share my thoughts today. So just in the beginning, I would like to make sure that my title is not misunderstood. By emphasizing the importance of rethinking here, I am not, I'm not saying that that what we're doing is wrong or what has been done is wrong. What I want to emphasize is that there is a need today to extend our hypotheses and working models and the approaches we're using on the basis of new findings that are highlighting really the complexity of this process and its role in neurodegenerative disease. So what you can see, this talk is mainly a call for embracing complexity. As a start with some disclaimers, my lab is currently engaged in a number of collaboration with industry. We have ongoing collaboration and receive funding from AbbVie and Idorsia. And I am also the founder, shareholder, and a CSO of a new spin-off company from the EPFL Indy Biosciences that is also active in the field of developing therapies and diagnosis for neurodegenerative diseases. With that, I come to the outline of my talk. The continuing failures of anti-aggregation drugs in the clinic has led to calls and demands to rethink the amyloid hypothesis and the role of neurodegeneration, of aggregation in neurodegenerative diseases. In this call, in this talk, I will not address this issue directly, but instead what I want to do is focus on making the case that we need to rethink how we're reassessing the role of protein aggregation in neurodegeneration. I will also address how oversimplification of the amyloid hypothesis and the growing gaps between the way we study aggregation in the lab compared to the way aggregation occurs in the brain has also contributed to these failures, and that embracing complexity is essential to advancing the field. I will then highlight some examples from our work that illustrates our approach to trying to deconstruct and reconstruct this complexity in the context of Parkinson's disease by looking at Lewy bodies. And then I'll discuss a little bit the implications for our findings in Parkinson for other neurodegenerative diseases. I will then go on to revisit the current hypotheses about the role of protein aggregation in neurodegeneration, with a bit of emphasis on the role of toxic species versus toxic processes, and highlight the complexity of anti-aggregation therapies, with the main message that I don't believe there is a single silver bullet that targets all aggregation mechanisms. I'll conclude with some concluding remarks, and then I'm really looking forward for the Q&A and discussion. I can't think of a more compelling evidence or a reason for that should force us to pause and rethink what we're doing than the fact that as of today, we don't have any approved disease-modifying therapies for these neurodegenerative diseases. And the clinical trials for drugs and antibodies targeting protein aggregation continue to fail in the clinic. It's very sad that Parkinson patients today are still receiving the same treatment that their grandparents received in 1965. And this has had huge impact, not only on the stocks of companies and the appetite of investment in neurodegenerative diseases, but on the lives of many people who are affected by this disease. And I'm constantly reminded by this, of this, by my interaction with Parkinson's patients, and especially with one of my close friends here, Benjamin Stetcher, who was diagnosed with Parkinson's disease at the age of 29 and is turned in and constantly reminds me of this quote, that the pace of discovery is slower than the pace in which his neurons in his brains are dying. And that emphasizes the urgency and the call that, for us to reassess what we're doing. And this is the message that we get from all the patients that we generally receive in our lab. So let's take a journey over the past and re-examine how we have been doing things and our success and failures. So today, if you look at the literature, you will find there are hundreds of drugs that have been reported to inhibit protein aggregation or disrupt the preformed aggregate and modify protein aggregation toxicity, but none has actually shown efficacy in the clinic. For more than 25 years, this has been happening. And if you look at the number of papers and amyloid and inhibitors, they continue to be in the rise. We have about almost 1,000 papers reporting on inhibitors of amyloid aggregation, yet we don't have any small molecule or drug targeting protein aggregates successful in the clinic. And there are many reasons to explain this, including the fact that these are complex diseases, that we still don't understand all the mechanisms. In many cases, it's the drugs that are the wrong drugs. Well, it's hard to argue with this many number of inhibitors. And there are issues with clinical trial designs, but also the preclinical models. There is really no model of Parkinson's or Alzheimer's disease. All the models we have, they model only one aspect of the disease, if they do. And then the issue is, is the amyloid hypothesis the right hypothesis? And what I want to, hopefully end with today is the idea that I don't personally believe that this hypothesis can be ruled out yet. I think there has been an oversimplification of the amyloid hypothesis. And I think it's important that we begin to accept that our reductionist approach has reached its limit, or that there are limits to the reductionist approach. So what's the problem? The problem today is the more we publish is actually the less we know. I've been in this field for 20 years, and I know of my colleagues in the field. Yet when we, the so-called expert get in a room in a conference, we still cannot answer the most basic questions, which is, what is the toxic species? What's the relationship between protein aggregation and neurodegeneration? You ask two people, you get 3 to 4 answers from different answers from each individual. Simple questions as what's an aggregate and what's an oligomer? This is one of the most frequent questions I get today, especially from colleagues in industry. What do oligomers in the brain look like? And the answer is we don't know. So the fact that despite all of these activities, increasing number of people coming to the field, increasing number of publications, we're still yet not able to answer the most basic questions. Now, I spent quite a lot of my time in the field, in my early years working on protein aggregation. And I spent, working on transthyretin, A beta, alpha, synuclein, tau, you name it. And as a PhD student, I spent about 10 years working on these proteins, not thinking about the disease, not thinking about the patients, mainly thinking about proteins and protein aggregation. So we think that the issue is really in the aggregates that you see here. Is it the fibril, the oligomers, the misfolded monomer? And our approach to understanding how we tackle or disaggregate or disrupt these is sort of aggregation centric. And that's okay if the goal is to understand fundamental mechanisms of protein aggregation. But if the goal is to understand how this process contributes to the disease, then we need to really move to the left and realize that the issue is in the tissue. In the sense that protein aggregation does not occur in a vacuum. It occurs in a very crowded environment of the cell that includes a complex proteome, it's lipids, and it usually involves, significant modification of these proteins, all of which appear to be cell type dependent because the environment changes in different cells. So the picture you see in the left is really what we have to deal with and what happens in the brain. So to what extent what you see in the right replicates the complexity that if we see in the brain is what I would like to go to explain in the next few slides. And then we will understand what's the implications for this. So this is, a persistent source of misunderstanding is really how we depict the role of protein aggregation in neurodegenerative diseases. So the image you see here is one that you commonly see when people introduce protein aggregation. And what people will say is that Lewy is, alpha-synuclein fibrils are the main constituent of Lewy bodies, or A beta or tau, and suggesting that these inclusions are mainly made of fibrils. And to some extent, this has been driven by sort of some of the early views we have in aggregation. For example, on here, you see an image by Fornow et al., in which shows the early images of Lewy bodies. And what you could see is a dense core irradiating filaments from the center. And it's very hard to ignore the abundance of the fibrils. But what you also see on the surface is that these fibrils are decorated. It took a while to understand what this core is. And you could see there is quite a lot of membranous organelles and other things that are in the periphery. So it was understandable why fibrils dominate. But the reality today is so different. Thanks to advances in imaging, this is a sort of advanced electron microscopy, correlative light electron microscopy, recently showed that Lewy bodies in the brain are not just made of fibrils. They're not just made of aggregates. They are complex mixtures of lipid, protein, small molecules, and disrupted membranes and organelles. So there is more to pathological aggregates than just the fibrils. Well, if you go further, you realize that these pathological aggregates don't come in one flavor. What you see here is a paper from Trzebowski's lab showing that the diversity of tau pathology that you could observe when you stain it with an antibody against phospho-tau. And the same for alpha-synuclein. You could see that Lewy bodies or Lewy pathology exist in different forms. On the right, you see these are the amyloid plaques. This is on the top what we commonly, you see when we talk about amyloid, but amyloid are equally diverse. I looked at the literature, and there is a very nice recent review by Larry Walker on this, and there are at least 11 different types of amyloid plaques that people have described. So what this suggests is that there is a huge degree of pathological diversity of these aggregates, which we're increasingly learning is actually is due to the fact that these different types of aggregates are actually structurally and biochemically different. So when we're talking about aggregates, they are not, there isn't one form. There is quite a bit of polymorphism, both, at the level of the protein and pathology. Another important thing that we've underestimated is the fact that many of these pathologies in neurodegenerative diseases co-occur together. And you see these in aged individuals, but you also see them in people who have Alzheimer's or Parkinson. There is quite a bit of overlap. In this study here that looks in front of you, they looked at 375 older adults, and what you could see, they could see what they call quadruple misfolded protein pathology. They see tau, A beta, synuclein, and TDP43. And there is evidence that suggests that these co-pathologies, when they occur together, there is an increase, sort of enhanced progression toward dementia, suggesting that they may act together. We know now that the interaction between these proteins occurs both at the molecular level, and they also have additive effect in the brain in the way they cause damage. And there is, suggestion now that tackling these diseases may require sort of strategies that tackle multiple pathologies. Now, one of the interesting things that you see in neurodegenerative disease is that almost all of these pathological modifications are heavily post-translationally modified. In fact, it is antibodies against the phosphorylated, ubiquitinated, or modified form of these pathologies that are the main tools that we use to quantify or monitor the formation of these pathologies in the brain. Yet, if you pick up any review in protein aggregation or neurodegeneration, I can bet you that in 90% of these reviews, you will find no mention of PTMs in the schematic that show the mechanisms of aggregation. And here we're not talking about the presence of one or just a few modifications. This slide shows the number of modifications that have been detected in alpha-synuclein isolated from the brain of patients with Parkinson's and different synucleinopathies. So you can see the distribution of different PTMs. And of course, the abundance of these PTMs vary. But what is very clear is that the number and abundance increases with increasing pathology. If you look on the right, you could also see the distribution of different PTMs that are observed on tau isolated from the brain. So this poses a sort of a major challenge in terms of giving this complexity and heterogeneity. How do we tackle this? The decision in some part of the community has been, because these things are too complex, then it's best to avoid them. And I will come back to that later. Also, when we deal with these protein links into aggregation, we usually think of them as one proteins, but the reality is that they're not. They usually exist in multiple isoforms, at least at the monomer level. And then usually at the aggregated form, you have an increased level of modified forms. Now, these forms are usually in equilibrium. We know that there is that beta-synuclein is a negative regulator of alpha-synuclein aggregation. We know that in different tauopathies, we have different level of aggregation of different isoforms. And we know that these proteins definitely interact together. Yet in most experimental approaches that we use, we still investigate these proteins one protein at a time. This is just a table. A beta is one of the famous protein used in the protein aggregation fields, and we now know that it exists as a mixture of different peptides ranging from 133 to 148. So this landscape of amyloid diversity has increased over the past couple years, mainly due to advances in cryo-EM. And now we know that amyloids come in many different shapes and flavors. And these shapes are reflected in different structure in terms of how these amyloid, the proteins are arranged with these amyloids. And studies from using animal models and others show that these different morphologies or different structures exhibit different seeding activity in different models, suggesting that perhaps the pathological diversity we see in the brain could be in part to the diversity of these aggregates of the fibrils that sometimes it's very hard to capture without cryo-EM. Now, the most interesting finding has come out recently, also in the past couple of years, thanks to cryo-EM, is the finding that amyloid fibrils we produce in the test tube are actually quite distinct and different from amyloid fibrils we isolate from diseased brain. Shown here are the cryo-EM structures in section B of all the synuclein fibrils prepared from recombinant protein. And the top is recent amyloid, is a filament, a structure of filaments that were isolated from a brain of an MSA patient by Michel Goudet and colleagues. And in this table, you can see the differences in the structure to make it easy. You can see it here in terms of the fold, and you could see it here as well. And what is very clear is that the amyloid fibrils that occur in the brain have a very distinct structure. They've also identified by mass spec a number of post-translational modification, including some novel modification. So this people have reacted differently to this. And many people are actually quite surprised, that this is the case. But I'm not, if you recognize from everything we knew before in terms of differences in modifications and differences of properties, one should not be surprised. We've seen the same thing in the tau protein, that generally tau in vitro is produced using heparin to induce tau fibrillization in vitro. And more recently, tau filaments have been isolated from the brain of Alzheimer's patients and from brain of patients with other forms of tauopathies, including Pick's disease. And the emerging picture is the same, that the tau filaments that you isolate from the brain are structurally and morphologically distinct from the tau fibrils that you see in vitro. And this is an example from our lab. These are recombinant tau fibrils that you typically get when you use heparin. And these are the tau filaments that you isolate in the brain. And you have to appreciate that a great deal of the drug discovery effort, antibody development, small molecule inhibitors were actually identified or designed on the basis of these type of aggregates. Now, we have made recent progress in our lab. I'm not going to talk about that today. That's a sort of, but we are actually now able to regenerate using recombinant full-length tau filaments that look very much close to that what you see in the AD. So it's very possible to begin to reconstruct that complexity. I'd like to end this section with this slide, and this is a recent study by Brad Hyman and colleagues, in which they collected they made oligomer preparation from brain cortices, from cortex of 32 Alzheimer patients, and then they took those oligomer preparation and used them in different cellular and animal model assays to look at their seeding activity. And then what they did is they characterized these preparation in terms of the amount of oligomers that are there and in terms of their phosphorylation or modification pattern. And what you could see here is that all of these are individual or being confirmed to have Alzheimer's disease. Yet you could see the large disparity in terms of how they behave in these assays and the large variation in the different types of oligomers and PTM profile they have. Although I don't agree with some of the conclusions in this paper, I think what their study nicely highlights is the complexity that there isn't a single thing as one Alzheimer's disease. And what is important for how we have, from an experimental point of view, is when we identify a target or we want to test how a candidate, the drug or a molecule is linked to a disease, we typically go and collect 2 to 3 Alzheimer's patients and we compare our data to control. And what this data shows is that the large variation here means that if one group took samples 14 to 16 and another group took samples 30 to 32, they would get completely different results. Emphasizing that establishing a link between in vitro and what happens in disease must appreciate the complexity and the heterogeneity of the disease. And thus one should include a large number of patient samples if we want to make sense of in vitro studies. I'm not going to talk a lot about the tools. I don't want to stay sort of on, but a lot of the hypotheses that we have are based on antibodies that we use to characterize pathology or use to characterize our models. And one case, for example, is the evidence for the presence of oligomers has been in vivo has been really driven mainly by the development of what people have called oligomer-specific antibodies. We recently completed a screen of 16 antibodies that were reported in the literature and commonly used to be oligomer-specific. You can read the paper, but what we found ultimately is that none of these antibodies is actually oligomers. All the antibodies that were supposed to recognize only oligomer recognize both oligomers and fibrils. And many of these antibodies were actually unspecific and recognize all species. And that, I think, again, should cause us to pause and rethink, sort of not only the experimental approaches, but how we validate and characterize our tools. Now, I hope I haven't painted a pessimistic picture of the field, because I just, it's important to really reflect on this, because personally believe that this is also an important contributor to the slow pace of progress in terms of developing therapies. Now, what I want to move on the positive side and show you and sort of that it is possible to reconstruct that complexity and not so that, you know, we don't have to be afraid of it and shy away from it, but instead embrace it. And I'll tell you this in the context of work where we've tried to reconstruct the pathology of more specifically Lewy bodies in Parkinson's disease. So I can't start talking about Lewy bodies without talking a little bit about Parkinson's disease. Many of you know this is a movement disorder. It's one of the most common ones. What people usually don't know is that there is more to Parkinson than the motor symptoms. There are motor and non-motor symptoms that are usually manifested in the disease, with the non-motor symptoms being manifested very early. The disease at the brain level is characterized by loss of dopaminergic neurons. And that's where we find this Lewy body pathology, which several studies, you know, we now know contain alpha-synuclein filaments within them. Now, what's interesting is that, you know, over the past few years, a lot of data are coming out from different groups showing that the synuclein and synuclein aggregates could be present in many organs and tissues outside the brain, which may actually explain some of these non-motor symptoms. So the picture we had of synuclein in Lewy bodies, as I mentioned before, was one that was dominated by fibrils from all the ultrastructural studies that you see in the 1960s. But actually, if you review this data very carefully and you go back through these papers, you find that in many of these pictures, there was always these Lewy bodies were sort of surrounded by these huge accumulation of these membranous organelles. In initial studies, these were labeled as contaminants. And in subsequent studies, people just cited their presence, but nobody thought that these are actually important or play a role in Lewy body formation. What we also know about Lewy bodies is that, you know, that synuclein is there is a number of post-translational modifications that characterize Lewy bodies and synuclein accumulation in the brain. It has been reported that 90% of synuclein in the Lewy bodies is phosphorylated. Lewy bodies are also heavily ubiquitinated and they can stain for neurofilaments. If you take amyloid-specific dye, you can stain them, suggesting that they're fibrillar, which is consistent with what we see by EM, but they also contain lipids. And in a sense, Lewy bodies is this complex mixtures of cytoskeletal proteins, modified synuclein fibrils, and membrane. So how do we deconstruct this? How do we start to look where does this process actually start? The way we started looking at this is we focused initially on the aggregation. Given the presence of the filament, we worked on the hypothesis that maybe alpha-synuclein fibrillization is actually what is driving this process. So it makes sense that, you know, to start this, that we would start by looking at how, you know, the mechanism of alpha-synuclein aggregation. I don't know why I'm not getting this. You know, sort of how alpha-synuclein aggregates and form fibrils. And given the abundance of PTM, the second question, a simple question, is how do PTMs contribute to this process? The original thinking was that given that synuclein is mostly phosphorylated, that phosphorylation, for example, at 129 and potentially ubiquitination, drive the formation of fibrils and potentially this conversion to Lewy bodies. And then the second question is at what point do these organelles come in? We know that synuclein interact with membranes, and membranes could actually contribute to the initiation and progression of alpha-synuclein aggregation. So there is a possibility that they could act early to drive the formation of Lewy bodies. And then, of course, these Lewy bodies are also full of other proteins. At what point these proteins come into the picture is not clear. So our goal was to try to dissect these individual steps, and at the end, try to correlate these individual steps with the neurodegeneration that we see in the brain. So obviously, we use the combination of different approaches, ranging from in vitro to primary neurons to use of primary culture neurons and animal models of synucleinopathies. So the challenge we faced in the beginning to do this, so the first question is, do PTMs enhance alpha-synuclein aggregation? And was which PTM do we study? There were a large number of PTMs that are identified in the brain. And the second problem we had is we didn't know the enzymes that regulate this, and we had no way of site-specifically preparing these proteins. So what we opted for is a chemical biology approach that was driven by taking our understanding of the modifications in Lewy bodies and chemically synthesizing these individual modified proteins, and then ask how do they affect aggregation and toxicity. Much of this work has already been published, so I'm going to go through it a bit quickly so we can go to the end. So the way we do this is basically by building synuclein one amino acid at a time. This is what we call total chemical synthesis of synuclein. In a sense, what we do is we take the big protein, cut it into small pieces that you see here, synthesize each one using solid phase peptide synthesis, and then stitch them sequentially, starting from the C terminus to the N terminus. That, since we're synthesizing each fragment by one amino acid at a time, we have a total flexibility to introduce PTMs anywhere we want and as many as we want. Just to give you an indication how this works, usually in a tube, we put the first fragments. And then, this fragment has an N-terminal cysteine. We have another fragment that has a thioester and a cysteine at the other end that is protected. And if you put this at pH 7, these two fragments ligate very quickly. We then simply lower the pH to open the cysteine at the end, add the third fragment, and you have your protein of interest, synuclein. And because we can add modification to each of these fragments, we can synthesize any modified form of the protein we want. Using this approach and others, we've actually been able to reconstruct the entire synuclein proteoform. And I think it's one of the only proteins for which we have remade in a homogeneously purified form every single form that we've detected in the brain, CSF, or blood. We have a library of about 46 pure synuclein, ranging from simple modification as nitration and phosphorylation to mono, di, and polyubiquitinated synuclein. So with all these proteins in hand, then we ask the question, which of these modification actually drives the process of fibril formation? And which one inhibits? So with the idea that if we have ones that drive the process, then we can go after the enzymes that are responsible and develop sort of those as potential therapeutic targets. What we learned is that most of the phosphorylation events that we detect in the brain actually inhibit or significantly delay rather than promote alpha-synuclein aggregation. Another thing we have learned is that the common approach of using phosphomimetic like glutamate or aspartate to mimic phosphorylation doesn't work. You can see here that the phosphomimetics behave like the wild type. And when you block phosphorylation, you actually enhance aggregation. So, and we've been able to replicate this in many systems now. Yet this approach is still used in biology. In many cases, sometimes you don't have a choice. What's interesting is that here you see that phosphorylation at Y39 delays the aggregation, but doesn't affect the structure of the fibrils. That's what we concluded in 2016. And we were wrong, because recent cryo-EM structure data showed that the phosphorylated form of the protein forms fibrils that are completely different than from the wild type, because this phosphorylation site falls in the core of the fibril, thus causing major rearrangement in the fibrils. So what we say here is that phosphorylation not only affects the kinetics of the aggregation, but the PTMs can also influence the structure of the final fibrils. And we see this with other modification. For example, here, nitration at the same residue has no effect on aggregation, but it changes the morphology and the structure of the final fibrils that we see. And the same as, when we have polyubiquitination. Interestingly, monoubiquitination inhibits the aggregation of alpha-synuclein, despite the abundance of the co-localization of ubiquitin staining with synuclein and Lewy bodies. So what we've learned is that from all of these studies is the following, is that it turns out that most of the modification that we see in the brain inhibit rather than promote aggregation, with the only exception of U and mainly C-terminal cleavage, because when you remove the C-terminal, you remove 11 charges that keeps the protein soluble. And what that leads us to think is that actually post-translational modification don't play a role in the initiation of the process. And they are actually not required for aggregation because synuclein will aggregate whether you have PTMs or not. And the working hypothesis we had was that PTM may be represented late event, that they're not important for the first step, but they're important for the regulating the transition from fibrils to the Lewy bodies we see in the brain, and perhaps regulating the interactome of the fibrils to form Lewy bodies. So now, if you want to test this, we looked at different cellular systems, and most of the existing systems based on overexpression of synuclein do not produce Lewy body-like structures. And we decided to try to explore this mechanism of both Lewy body formation and role of PTMs in the context of a neuronal model. And we liked neuronal models or cellular models because you have the ability to interrogate both the aggregation process and cellular response at a very high detail level by doing transcriptomics, proteomics, and advanced imaging. So our first goal was to try to generate a model where what we call is a neuronal model of Lewy bodies, or a Lewy body in a dish. Initially, we started to approach this by trying to simply overexpress synuclein in neurons. We faced the same problems as others in that sense that it didn't work. Synuclein, when you overexpress in neurons, you do not get inclusions. We later found out that one of the problems you don't get inclusions in neurons is because when you overexpress the human protein, you have the mouse protein in the neurons, and the mouse protein blocks the aggregation of human alpha-synuclein. So if we now do the experiment where we overexpress wild-type human alpha-synuclein in wild-type mice, you have no inclusions. But if we do this in alpha-synuclein knockout, in the primary culture from alpha-synuclein knockout mice, then we see abundant inclusions. When we go and look at these inclusions by electron microscopy, we can find that these inclusions appear to have some filamentous structure, but they don't have that typical radiating and dense structure that we see in Lewy bodies. So this model is able to produce fibrils that have this phosphorylation ubiquitin staining, but it doesn't have the organization or ultrastructure properties of a Lewy body. So then we decided to say, okay, maybe we need something to initiate this process. And the idea here was we go to wild-type neurons, we add fibrils that are prepared in vitro, and by doing this, we can initiate the aggregation through a seeding mechanism where the endogenous protein is then recruited and forms fibrils rapidly, which then go on to do Lewy bodies. This model has already been established by Virginia Lee lab, and it's been replicated in different cellular systems all the way to in vivo, and it actually works. If you take preformed fibrils, add them to neurons without overexpression, then you could see that they go on to form these filamentous structures. So the concept of the seeding works. And when we do this in the lab, it actually works. So these are different prefibril seeds from recombinant protein. We sonicate them to make it easy for them to get into neurons. And what you could see is when you add them to neurons, we looked at the same time points that people usually look, 11 to 14 days. You see the formation of these filamentous structures that stain with phosphorylation 129. They are ubiquitin positive. They bind thioflavin S and other dyes. So they have all the characteristics of fibrils. And then when we look by electron microscopy, you can see these long filamentous structure that are there. But we still did not see Lewy bodies. So we were curious as what is the, why we cannot produce these Lewy body-like structures. Sort of we learned that, there are no secrets that time does not resolve. And if you know that everything takes time. And so we kept trying to, use these seeds. But what we realized from an in vivo experiment that we've done is when we inject these fibrils in the brain of these mice, within 30 days, we don't see Lewy body structures, but we see this neuretic accumulation of synuclein aggregates, which over time can convert into this condensed structure. Then what we realize is maybe the only problem we have is that we were not waiting enough. And indeed, when we go to the primary cultures, now we go, this is 7 days, you see the filaments in the neurites. And then if you go to 14, you see these filaments mostly in the cell bodies. But then if we wait to 21 days, you begin to see the conversion from these filaments to these globular structures that look like Lewy bodies. So we were very excited in the sense that we now have what we call as a Lewy body in a dish. But the question is, do they actually resemble Lewy bodies that we see in the brain? And for this, we did extensive correlative light electron microscopy with the imaging facility here, Graham Knott and colleagues. And these are the images. So you could see in the beginning of this process where we see accumulation of synuclein in the neurites, you see mainly single filaments, alpha-synuclein fibrils. By 14 days, we see major changes in the sense that the fibrils are getting remodeled. They begin to get shorter and clump together. And you begin to see the recruitment of membranous organelles around the fibrils. By 21 days, you have these, when you have these inclusions, you find the formation of these inclusions that are full of fragmented fibrils, mitochondria, autophagosomes, lysosomal structures, and damaged membranes. And you could see a better structure of this here. And we could stain with different organelles, and we can see the accumulation of these. So this seeding model is now sort of reproduces many of these cardinal features of Lewy bodies. When we look when we do sort of isolate these inclusions and do the proteomics of these, we find, for example, 20% of the proteins that are usually found in Lewy bodies, we find in these inclusions. We find many proteins, high concentration of proteins that are linked to the protein quality control machinery, mainly linked to proteasome, autophagy, chaperone-mediated autophagy, suggesting that this process sort of causes major disruption to the protein degradation or protein quality control machinery. Suggesting, again, pointing that the process of Lewy body formation, the contribution to the mechanism of neurodegeneration may go beyond that of simply aggregation. Okay. So this is a comparison of what we see in this neuronal model and what you see in these synuclein Lewy body structures from substantia nigra of Parkinson's disease patients. We call it Lewy body-like because Lewy bodies take a long time to form, but we were very excited about this. So with this now in hand, the question now we felt we have a model in which is unique in the sense that we can model the two key steps involved in the process. We can model, we have between 7 and 14 days, we only see fibril formation. So we can ask, what regulates this process and how this process contributes to neurodegeneration. And then we can also look at later times and look at, what is the contribution of the transition from fibrils to inclusions in terms of neurodegeneration, or which of these processes is actually responsible for the neurodegeneration. I'm going to go through this quickly to summarize it, just to say that at 7 days, when we only have fibrils in the neurites, we have all the markers of Lewy bodies, that there is immunoreactivity to P62, phosphorylation, ubiquitination, but there isn't yet C-terminal cleavage of the aggregates, and there is no degeneration or any signs of dysfunction. At 14 days, where we still have fibrils, we don't have inclusions, but what we have is the beginning of coalescing, of recruitment of organelles. we see the same, all the markers. We began to see an increase in C-terminal truncation of the protein. We began to see a remodeling of the fibrils and ultrastructural changes that signal the beginning of the formation of these inclusions. And we began to see accumulation of large number of proteins. In terms of degeneration, we don't see significant degeneration, no mitochondrial dysfunction, but some synaptic impairment and early cell death events, signaling events. When we go to 21 days, where we have the transition is complete to inclusions, this is where interesting things began to happen. We see an increase in the number of post-translation modification and increase specifically in level of truncated synuclein. We see a remodel, the formation of these inclusions, but when it comes to cellular homeostasis, we see major dysfunction in mitochondria from respiration to decrease in membrane potential, and also decrease in the level of many mitochondrial proteins. Same when we look at synaptic dysfunction, we see many signs of synaptic dysfunction at many levels, and also cell death. What this suggests is that it is not the initial processing for fibril formation that drives neurodegeneration, but rather the transition from fibrils to inclusions and the interaction between these fibrils and other proteins and organelle that drives this process. So as Albert Davis wrote in one of his commentary in our paper, it's beyond synuclein. It's the organelle accumulation that could be contributing to this process, and we're trying to figure out now specifically how. So what we've shown is that alpha-synuclein fibril formation is not sufficient to induce neurodegeneration. So everybody always asks me, what about the oligomers? And I can say that we do not detect accumulation of oligomers in this model. And that's because when you add a seed, they sort of hijack the process and we don't get oligomer accumulation. All the monomers goes into the growing end of the fibrils. What we show is that the process of Lewy body formation, rather than a specific aggregate or the fibril in this case, is really one of the primary drivers of neurodegeneration in this seeding model. What this suggests is that the damage is happening during the process, which means once Lewy bodies are formed, removing Lewy bodies may not be protective. And this reminds us of all the experiments in the amyloid hypothesis in the Alzheimer's disease, where people show that if you use antibodies or drugs, you can remove preformed plaques, but you get no cognitive benefit. And that's because maybe the damage is happening during the process and not necessarily by the final product you see. So if our hypothesis is correct that this process, then would one expect that the neurons bearing or that show Lewy bodies are the ones that die. And it's nice when you think of an experiment and then you realize that somebody else has done the experiment. And in this nice paper, what the authors did is basically they used a similar model, except in this case, they did it in vivo, where they expressed alpha-synuclein fused to GFP and then added preformed fibrils to these mice. And they can show that the same thing as we see in our model. Initially, neuretic inclusions, which then began to go through stages that are accompanied by increased compaction of the fibrils, which they analyzed by FRAP. And you could see here, this is an image of two neurons, one that has an inclusion and one that does not have an inclusion. And if they monitor over time, they found that neurons that have inclusions die, whereas the ones that don't have inclusions survive longer. So this is again supporting the idea that the process of Lewy body formation could be the one that's driving this. So now the question now is, what do PTMs do in this process? And, so what we thought now with this model, we can ask this. So what we found in this model is we start with these single fibrils, then we have this lateral association with the fibrils and then formation of this. And this process is accompanied by increase in the level of truncated synuclein. And a recent study showed that the Lewy bodies, which confirm previous studies, are made of these multi-layered structures in which the truncated species go in the middle and the phosphorylated species are found in the periphery. So what that's led us to suggest is that the C-terminal truncation, in this case PTMs, play an important role in the compaction of the fibrils. Why is this the case? Because C-terminal part of synuclein has 11 negative charge, 12. And therefore and it's very flexible and exposed in the surface of the fibrils, thus making it difficult to pack the fibrils in Lewy bodies. And indeed, if you make different truncated form of synuclein, you can see that as soon as you begin to remove a significant number of charges, they begin to clump together. So the hypothesis is that when these aggregates form in the cell, it's very difficult in the neuron to pack them into Lewy bodies. The only way possible then is to remove the C-terminus, and that drives their accumulation and formation of Lewy bodies. An alternative hypothesis is that binding to other proteins could neutralize that charge and also drive this process. It's very difficult to test the role of truncation, and the way it has been done is people make a truncated protein and a full-length protein. But in this case, the truncated protein aggregates in a very different mechanism. So what we try to address this question is by actually engineering a form of synuclein where we actually include a photoactivatable group here. That's when we activate with light, actually cleaves the C-terminus. So it's a light-activated cleavage of synuclein. And what we show that if we make fibrils from this protein, then you have the C-terminal part of the protein here is intact, and we make mostly individual fibrils. As soon as we shine light to cleave the C-terminus, these fibrils begin to clump again. So it's a very nice way of illustrating the role of post-aggregation PTMs. And this is sort of what we have learned now about the PTMs, is that they're complex and play a role at different stages of Lewy body formation. So if we look at truncation as one example, and I don't have time to discuss the others, you can see that if you cleave the synuclein monomer, you produce a truncated form that has less charges in the C-terminus, and that drives its aggregation to form the fibrils. What we have also learned is that when we add these preformed fibrils to neurons, almost within 6 hours, they completely get cleaved by calpain and others, producing fibrils that lack the C-terminus, which then grow by the addition of endogenous protein to form newly formed fibrils from the full-length protein. Interestingly, is once these newly formed fibrils are formed, the PTMs come in. The first PTM we usually see is phosphorylation at 129, which then signals ubiquitination. And ubiquitination signals the recruitment of P62 to try to target these for degradation by autophagy. And once these modifications are there, these fibrils get modified again to facilitate their lateral association and packing. We're still trying to dissect now, the role. We believe that these PTMs are not markers of pathology, but they're master regulators of this process because they regulate the interactions. For example, you heard in one of the recent lectures in this series that HSP bind to synuclein and disaggregate synuclein. It's interesting that HSP70 binds the chaperone complexes to the C-terminal fragment of synuclein. If you remove it, then there is no disaggregation activity. And what you could see here is that this C-terminal part is the part that is heavily modified with phosphorylation, but it's also the part that gets cleaved, which means in the context of C-terminal synuclein, most chaperones becomes ineffective in disaggregating the protein. So now, what does this mean? I close this what does this mean in terms of therapeutic strategy? Well, the most effective therapeutic strategy to deal with aggregation is to prevent aggregation from the first place. And the way you could do this is by, targeting the monomeric protein, because if you do that, you block all subsequent steps from fibril formation to Lewy bodies, and then you block sort of the pathology spreading from neuron to neuron. To do this, you need to stabilize the monomer, and this has not been proven to be easy, or you need to lower synuclein levels. And it's very difficult to do this when you don't understand a lot about the biology of synuclein. But what we have also showed is that PTMs can actually regulate synuclein levels. So we showed that synuclein is phosphorylated by polo-like kinase 2. It's also phosphorylated at 39 by c-Able. And in every case, the phosphorylation sort of inhibits or delays aggregation significantly. But what is interesting too is that PLK2 phosphorylation of synuclein targets the monomeric protein for degradation by autophagy, whereas inhibition of c-Able kinases targets the protein for degradation by proteasome and autophagy, suggesting that regulating PTMs could provide a way of regulating synuclein levels, but activating these kinases is not easy, so one needs to look at upstream pathways for regulating the activity of these kinases. And we showed in vitro that this translates into lowering toxicity. Now, the problem with doing that is not enough, because when you get a Parkinson's disease patient, these people already have pathology in the brain, because the disease starts developing 10 to 15 years before the symptoms are manifested, which means that these aggregates are already moving in to different parts of the brain, are already propagating from one neuron to another. And what that calls for is that you need now a different strategy that interferes with this propagation. And the common strategy that is used today is to develop antibodies to target the species that are secreted by neurons, because if you can do that and block their entry, then you can block the propagation. But the main thing here is you can block the propagation, but if these neurons continue to still produce the aggregates, it's still going to be an uphill battle. An alternative strategy is another complication to this mechanism is the fact that there are different types of aggregates that are propagating. So it may not be possible that a single antibody can target all these different species that are propagating from neuron to neuron. So an alternative strategy would be to try to harness the power of the degradation machinery. We have shown that this could work by autophagy, and where you can enhance degradation of the fibrils, and that could lead to lowering fibril level, and again, allows you to target this process. Because in neurodegeneration, we're typically dealing with people at different stages of the disease, and most of the time, people who already have pathology, it's going to be very difficult to use a single approach to target this. And I believe this is partially or a major factor for the failure of current trials, because we tried to target amyloid without tau, or tau without amyloid, or one pathology versus the other. And this is why I think there is no silver bullet to tackling all aggregation driven pathogenic mechanisms, and this may require the development of combination therapies that target or different mechanisms on this pathway. And this has already been suggested. This is from a nice review by Eliza Maslia, suggesting that it's important to take this into consideration, and maybe we need to target different proteinopathy, different protein aggregates in the brain, but also target aggregation and other mechanisms that are emerging to play a role in these diseases, like in neuroinflammation, the immune response, and metabolic function. So I finish with these slides on sort of where we see the future directions for us. One is we need to go back to basics and really understand what is the molecular structural basis of pathology in the brain. It's been 100 years since we identified these pathologies. We still don't know what they're made of. We still don't know what is the basis of their diversity and to what extent it correlates with clinical heterogeneity. We need to revisit the role of PTMs in regulating clearance, explore how to harness the power of the degradation system for therapeutic purposes, and most importantly, not to focus only on aggregation, but explore the interplay between protein aggregation and other mechanisms that contribute to the development of neurodegeneration. And I see the future definitely in combination therapy. I end with thanking the people who've contributed to this work. I've highlighted their names in the slides. They are highlighted here in the arrows. This is the old picture of the lab but most of the people who contributed to this work are here. This is a new picture, and this is the team at ND Biosciences who are helping us translate many of this work and basic mechanistic studies into therapies in the future. And thank you for your attention. Happy to take questions.
Thank you very much, Hilal. Let me extend to you also on behalf of the other attendees a round of applause, at least a virtual one. And it's time for questions. So as usual, raise your virtual hand and I will unmute you one by one in the order where you raise your hand. So the first hand that was raised was from Pierre. Pierre, you can go on.
Yeah. Well, great, really great talk. And I learned a lot. You made me really feel guilty not to read enough of your papers. I was wondering about this challenge of yours to identify all these species in vivo, the ones that are more toxic than others, etc. What goes wrong? And in a way, coming from the chaperone world, I say this is the challenge of the chaperone and eventually the proteasome and other system. How this system can efficiently, apparently, identify those species in youth, and what goes wrong later on when the disease develops. I was wondering if knowing these intricacies of the different structures and the formation of the different fibrils would help to understand how the system in the cell naturally already is eventually able to identify and kill them when they're small.
Yeah, I think, it's very clear, that sort of, I go that the sequence plays an important role on this. And I think, in my view, that the reason we find these PTMs and these aggregates in the brain, they're not there just to make them look good. They are there to signal events and I think related to interacting. It's very clear for us, that I don't understand how the chaperones recognize specifically the fibrils because, the C-terminal part of synuclein is displayed flexible in both the fibrils and the oligomers and the monomer, but maybe the density or the distribution of these in the fibrils combined with the PTMs is sort of what signals this interaction. Now, it's very clear when these things are formed that the chaperones are recruited. Because when we look at the proteome of this inclusion, we see a large number of chaperones that are within these. We don't, and that tells us that somehow they've been recruited to try to clear these or process these aggregates.
I was just wondering, maybe in this way related, I mean, clearly with the HSP70, which you mentioned, we and others have observed, of course, that HSP40 is more the mediator of the binding of 70, and there is no binding of 70 without 40s. And these can be very different and vary a lot in mammalian cells. And I was wondering if the quality of those substrate recognizers for 70 would be indicating what goes wrong in those Lewy bodies. The composition, Nilegoda and Bukau showed that, B1 and A, whatever, they are very important for this aggregation. So I was wondering if you can see a difference of balance of those in the Lewy bodies, for example.
We are trying to do this now, and the way we're approaching this is we would like to start from the human part. So we're doing some global analysis and proteomic and gene expression studies that have been done in Parkinson's disease patients and also in specific tissues that are affected in the disease and comparing those to the model. We don't want to jump and speculate too much based on this neuronal model, but if we see a strong correlation, because we will get many hits and we want to narrow this by focusing on ones that overlap with the human pathology. Yeah. But we have that data and we would be happy to look at it. I think what's interesting about that study is our study shows that if this C-terminal part of the synuclein regulates many of its interactomes, right? And what we show is that when it's cleaved, those interactions are gone. And what they have shown also with HSP70, that it does not disaggregate the C-terminally truncated form of the protein.
Yeah.
So the it's still very interesting. The question, unfortunately, has not been answered because we show that neurons that have Lewy bodies die. But the level of maturity of the Lewy bodies we see in these models is not the same as what you see in the brain. We have to be honest about that, right? So you could think about this in two ways. You could say that Lewy body process could be protective if it's efficient. In other words, if the fibers are formed, they're sequestered very quickly, and you remove all these damaged organelles, then that could be a protective process. But if for any reason that this process gets stalled or slowed down, then you have increased accumulation and disruption of cellular homeostasis and damaged organelles that increase the toxicity. And the other thing we're thinking about now, once these fibrils, can we sort of prevent Lewy body formation or degeneration by simply enhancing autophagy and degradation of these aggregates? It opens up many different ways for trying to tackle the relationship between aggregation and degeneration, and also to explore different therapeutic approaches to modulate the process.
Thank you. Great. Thank you.
Satyam Tiwari. Satyam.
Thank you. Can you hear me?
Yes.
Yes. Thank you so much for the amazing talk, amazing data, eye, ear, brain openers, actually. So my question is for the Lewy bodies. Do Lewy bodies are specific to only neuron cells? This is the question, because now we know that protein misfolding diseases are there at many organs of human body.
Yes.
So I was trying to understand, can we study the Lewy body formation in other tissues?
Yes. So let me answer this in different levels. So there is increasing evidence that you can find synuclein in many tissues in this, in the body, in the skin, in the gut, submedullary gland, many other places. The level of characterization of that pathology, in my opinion, has been poor, in the sense that most of these studies, and we have a review we're writing on this, have characterized pathology in the sense of phospho-S129 immunoreactivity, right? They equate the presence of phosphorylation signal with pathology. And there is data from synuclein aggregation in the appendix showing that you don't have, you have phospho-S129 without aggregation. So one has to be careful about that. The impressive evidence that is coming is that if you take a CSF of a person with Parkinson's disease, and you take 5 microliters of that CSF, you add it to a solution of synuclein monomer, you can actually induce seeding effect. And the level of synuclein in the CSF is very low. You can do this with brain homogenates, and a recent study showed that you could do this with the skin biopsies. So the assumption here is that the presence of these seeds in very small amount can be detected using these amplification assays, which allow you to amplify the aggregate just like a PCR reaction. So these data are suggesting that there is some pathogenic form that can be detected in sort of peripheral fluid and in skin biopsies. And this is, actually, in the case of CSF, you can diagnose, you can tell the people who have Parkinson from those who don't have Parkinson with very high accuracy just by using the seeding assay.
Exactly. That's why I asked this question. Do we detect it before from other tissues of human body, the pathology of the disease? Thank you so much.
Indirectly through these amplification assays, yes.
Yes, thank you.
Okay, next question is from Safak Air. Safak, you can talk. You just have to unmute yourself. Yes.
Hi.
Hello.
Thank you for the great talk and also inspiring work. So I definitely agree with you that prevention is more important than removing the Lewy bodies. So the seed should be, nipped from the beginning. But then something that I'm kind of curious about, like, what do you think about the translatability about your Lewy bodies in the dish model? Because in the beginning, as you also mentioned, tissue matters, and hippocampal neurons, are they also a progressively dying population in Parkinson's? Or like, how do you think that it would be transferable to the dopaminergic neurons if we were to develop a therapy based on hippocampal neurons, because those two neurons have different proteomics and signaling mechanisms?
I completely agree with you. Those are all very valid arguments. For us, we see this as a starting point. That's why at the end, I still called it Lewy body-like inclusion in a dish. We are exploring, we are doing two things. I mean, we have three goals. One is to transform this from a neuronal model into a humanized model, preferably sort of an IPS-based model. Two is we're looking at the cell type dependence. We are already looking at dopaminergic neurons, cortical, hippocampal, and even alpha-synuclein seeding and handling of aggregates by glia cells. And this work is ongoing. And third, we are not focusing on neurons. We are actually revisiting the human pathology in the brain. And we just received a grant from Michael J. Fox to do this. So one of the things we discovered here, for example, is that this abundance truncation of alpha-synuclein that happens late in the process. And this actually has already been identified in the brain. We know that truncations are among the most abundant modifications. But if you look at the antibodies that are being used in the field to stain for synuclein pathology, or even to target synuclein, they're mostly C-terminal targeting. So we are going back, and we have proposed a sort of a new way of doing pathology into sort of what we call molecular pathology, that we need to revisit pathology at the biochemical, structural, and organizational level. And so I think that any work we do in models have to be driven by what's happening in the brain. But it has taken us some time to get here, and we're now teaming with the right people to do this. So very valid points, and all of this is the next step, I think, for us.
Okay, so
thanks for the answer, and best of luck.
Thank you.
Thank you. So I will, before giving again voice to one question, let me pick a few questions from the Q&A section that were written. So the first one is, does alpha-synuclein that carries mutation end up in different kind of aggregates in the PD patient brain when compared to sporadic PD cases?
Yeah, I don't, most mutations in terms of pathology, show the same type of pathology. There is a new mutation, one thing that people need to remember is that synuclein aggregation is not only linked to Parkinson, it's only linked to other neurodegenerative diseases. Dementia with Lewy bodies, multiple system atrophy. And in these diseases, it occurs in different types of cells. For example, in the MSA, it happens in glial oligodendrocytes, and these glial inclusion have a very different organization than what we see, for example, in dopaminergic neurons. Most of the mutation, I think they show some. There are patients who are affected by some mutation that show more diversity in pathology, but there is quite a lot of overlap.
Okay, thank you. So another question that I will try also to frame it in a slightly more general way. The question is the PTM of nitration on tyrosine thought to be possibly reversible? And in general, to what extent all these PTMs are reversible?
Very good questions again. Nitration, I'm not aware of how to reverse nitration in the cell. If somebody discovers an enzyme that does that, I think, denitrates, then they're up, they should start their own company. A lot of these modifications, it seems that these modifications are much more dynamic when they happen in the native state of these proteins, because you can detect them, but they don't seem to accumulate. And that makes sense because a lot of the reversible modifications are there to usually regulate fast events. So you expect phosphorylation, dephosphorylation. They seem to accumulate in the pathology, and the level of dephosphorylation, because they accumulate quite a bit, we think that the extensive dephosphorylation at the fibril level is much less than what happens at the monomer level. What has been difficult to do, because using chemical synthesis, we can synthesize the monomeric modified protein and look at their aggregation. But I just told you that a lot of these modifications also take place after the fibril. So what we have done in the lab over the last 2 years is we've actually also developed method that allows site-specific post-translational modification of the fibrils. And now we can actually make the fibrils induce site-specific modification at one or multiple residues and beginning to answer the question, how does that affect seeding and whether that could actually influence fibril remodeling? We have not, people have shown with tau and other proteins that if you isolate them from the brain and you dephosphorylate some of these aggregates, they undergo major remodeling and change. But we have not done this for alpha-synuclein from the brain. So the short answer is rapid dynamics at the monomer, very low rate of reversing PTMs once they happen in an aggregation state.
Okay, thank you. So let's go back to boys. Dr. Rajiv Sivadasan, you can ask your question. Rajiv? He's not there anymore. So let me just pick another question then from the this long one from the Q&A. So 2 questions actually in one. In Lewy body in a dish model, how similar is the seed and the induced fibrils at day 21 in terms of the structure? And the second question, how was AD tau-like fibrils generated with recombinant tau achieved in vitro?
Yeah. Okay, I'll go to the first one because it's easy one to answer. The second one is I can explain the concept to that. So how similar are the fibrils that are produced in the cell to those we isolate from in the Lewy body? So we actually, I had prepared this slide in saying that this is, in our view, I started by asking you, how do we bridge the complexity of what happens in the brain and the simplicity of what happens in vitro? And actually our view is to do this is at the fibril level, is by isolating fibrils from these Lewy body in a dish, or generating them using the chemical approach that we have. We are actually in the process of doing this. So the idea is for us is we know, if you use recombinant fibrils, they don't have any of the modifications, right? So I don't think they're ideal for investigating how chaperones or other systems clear alpha-synuclein aggregates in the cell. So if we want to do this, the ideal is we can either isolate fibrils from the brain, and it's very difficult to get enough Lewy bodies from the brain to do this. And it's very difficult, to isolate enough to do this or at a scale to enable research in different labs. We're trying to develop now efficient protocols that will allow us to do this from cells. And our next goal is to isolate it and then perform cryo-EM structure on these. And hopefully that will tell us to what extent are the fibrils that we produce in these neurons similar or different from those we produce in the brain. Now, I have to caution about something because this data showing that fibrils are isolated from MSA patients are different from what we produce from recombinant has had a very bad reaction. And it's actually influencing decisions when it comes to funding and paper acceptance, because people are beginning to demand from people is, why are you working with recombinant protein? You should be working with material from the brain. And I've seen grants being sort of rejected because of this. And I think this is unfair because First of all, we've only had one structure from the brain. Second of all, we've learned so much from working with these recombinant proteins that a lot of what we know about neurodegeneration is driven from this. What we need to do instead is two things, is to make these brain samples available before we demand people do it, and for people who are doing in vitro to begin to move and embrace this complexity, at least at the biochemical level. To the second question, which is, the tau, I'm not sure I can disclose this now, but I can tell you that it's recombinant tau. It's one of the technology that we're using in ND Biosciences, and it requires a seeding, sort of the development of a template that allows us to reproduce that misfolded structure in vitro. But it works. Sorry, I cannot say more.
Okay, thank you. There are no more questions. There were still a few, and maybe I can just pick this one that it's intriguing. Do you think that different PTMs can overlap or take cover each other? So essentially, what is the interaction in a way between PTMs? Is there a logics in the PTMs and so on?
Yes. So if you look at synuclein, tau, or Huntington, or any of these proteins, it's very interesting that the PTMs are not distributed randomly. In other words, in synuclein, if you look at phosphorylation, oxidation, C-terminal cleavage, all cluster within 10 to 12 residues in the C-terminus. What that suggests is that there is a strong crosstalk between these PTM, and we have shown this. We have published already on this. And so I think there is first that crosstalk is very important, and it acts as a molecular switch between different states. The second phenomena is competition between different PTMs. We believe that's a second level of control. So we don't know whether they overlap. Yes, we know that these PTMs, several of the PTMs co-occur together. And there is a big problem there in the field, because when you look at the map of synuclein or tau, you see that there is 84 phosphorylation sites of tau. And that actually has had a big negative impact, because when we started to work on synthesis of tau, which we have accomplished, people say it doesn't matter if you can make it, because where are you going to start? But that picture is inaccurate, because the way people draw that is they isolate tau from the brain, and then they do mass spec. And then they map all the identifications they find from the different molecules that come from different cells, that come from different environments, into a single line. We need to go back and revisit the PTM patterns of these proteins with an emphasis of mapping exactly what you just said, which is which of these PTMs actually co-occur together. We know there are a few that do, and we know that they can talk between each other, and the presence of one versus two has different effects. Ultimately, I believe that the PTM code of these proteins is not a single letter code, but more of a combinatorial code. There are PTMs that target two different regions, and there are PTMs that control interactome. So the short answer is yes, we believe that crosstalk exists, and that competing PTMs are part of the mechanism of regulating the properties of these proteins and their PTM codes. How do they do this, or under what conditions they do this? This is something we have to look at and work at in the future. One of the first steps we took toward this is mapping. We have mapped all the kinases that regulates the phosphorylation of tau, Huntington, and synuclein, but that's only for phosphorylation. We need to do this for other PTMs as well. And then we have the tools to begin to ask these questions.
Okay, thank you. Do you feel like taking one more question?
No problem.
Okay, then from Shahrazad El Amri. Shahrazad, you can talk. You just had to unmute.
Thank you very much for giving me the opportunity to ask this question, and thank you to Hilal Lashuel for this impressive talk. Okay, I have a question more on the clearance systems. So you merely mentioned ubiquitin proteasome and autophagy as the way to degrade aggregates. What about other proteolytic machineries, and is there a dependence on the species, the aggregate species, or what about the microenvironment of the that is present in the Lewy bodies? Indeed, there is some cocktails of protein, including other proteases that may contribute to the degradation of the aggregates.
Yeah, I think this is a very good question. It's good to see you here. So, you know, in the context of our work, you know, we have not looked into this in details, right? We have looked at autophagy because we've been working on autophagy in Huntington, but we're trying to map this right now. And it's very likely that multiple degradation machinery are at work here. Whether they act at different stages of the process, that's something it's not clear. But it's something that I think one advantage of the system, and hopefully sort of other versions of this model, is that, you know, the fact we can dissect the fibrillization from the Lewy bodies. So we can, you know, we can go to the stage of fibrils and say, let's upregulate this degradation pathway versus this one, and see which one is most effective. We do have, from the transcriptomic and proteomic signature that points at, you know, sort of different mechanisms of degradation, but it's not one. You know, there are multiples. And as I said, you know, our strategy for following up on these different leads that we have gotten from this model system is really to first revisit what's in the human and begin to, you know, to prioritize the ones that correlate more with the pathology. So the short answer to your questions is all of these are possible, but I think we have a very good system that we can begin to dissect this. And it's only one model. Another thing that we've learned from the system, and we've learned is two things. Is one, not all, you know, amyloid proteins are the same. You know, so Huntington has a very different mechanism of aggregation. You know, we have a paper in bioRxiv that outlines similar studies to what we just did with Synuclein with Huntington. And one aspect that, you know, people have maybe, you know, we're focused on aggregate and degradation is that what we've seen is that this process usually happens in phases. So in Huntington, usually you have a collapse of base driven by the polyglutamine domain. And then you have the formation, you know, continued aggregation and active recruitment of proteins. Many of these proteins are functional proteins and important for the cell. From important transcription factors to proteins that are important for, you know, regulating protein homeostasis. So the process of it's not just the toxicity, it impacts on organelles and causing organelle dysfunction, but also the fact that a lot of important proteins that are recruited, which could disrupt protein homeostasis and cellular function. So it is not a single mechanism that this process. That's why, in my opinion, you know, that we need to balance our view and how we look and try to target this pathway from focusing on a species to focusing on the process. Because once we start looking at the process, it opens up many different possibilities. If you're focused on oligomer or fibrils, it's a very narrow way of looking at this. And I think that's sort of our view.
Okay. Thank you very much. But you have also the importance of some biomarkers that are proteases that are under regulated in this process that may be involved in the clearance of these aggregates, pathologic aggregates.
I agree with you. As I said, you know, the other thing that's good about this model is we have it characterized at ultrastructure, biochemical, and omic level. We have all this data, and we will be delighted, you know, to collaborate with people on very different levels, you know, leveraging the capabilities we have or the model we have. we cannot do it alone. Only to move forward.
Okay, I totally agree with you. So I will probably recontact you in the next weeks.
Sure.
Thank you very much. Thank you.
I think that this has been really an exceptionally interesting talk. Thank you, Hilal. Again, I extend a round of applause, and the recording will be online as soon as Zoom processes it and makes it available to me. And also by tomorrow or Thursday, you will get the announcement of next week's seminar. And so with this, thank you, Hilal, again. Thanks all the attendees. And if you have more questions, I think that they can address to you directly by email, I think.
Thank you very much, Paolo. Thank you all for the interactive and stimulating session. Have a great day.
Thank you. Bye.